All right, we should get started this biology seminar. So in the name of the Biology Department Committee on Seminars, I welcome you all to today's research talk. Before we get to that talk, I want to remind you of upcoming seminars in that series. On March 10 is our next seminar. It will be a seminar on career development, and Merlin Benner is going to talk about what it takes to be a biologist and wildlife specialist. Our next research seminar is going to be by Dr. Sarah Wright from the biology department, and she's going to talk about the genetics of plants. So you can see here the whole set of seminars. These announcements are all over Science Hall, so you can find ways to connect. So today, it is my pleasure to welcome Dr. Archana Kumari. And before I start talking about her, I'm going to start recording. Although it seems that somebody is recording already. So I don't have to do it. So it is my pleasure to welcome Dr. Kumari. She is an assistant professor in the Department of Cell Biology and Neurosciences at the School of Osteopathic Medicine here at Rowan University on the Stratford campus. Dr. Kumari got her BS degree from the University of Delhi in India, and then she got her master's degree in the Awadesh Pratap Singh University, also in India. And then she did her PhD at the University of Gujarat, also in India, before coming to the US as a postdoc to the University of Michigan. And then that's as a postdoc at the University of Michigan that she started to work on the topic that she's going to talk about today, which is taste, tongues, and hedgehogs. We're not going to talk about hedgehogs as the animal, we're going to talk about hedgehog as the protein, as I was told. So sorry for all the biologists here, we're not going to see a lot of little spiky animals, I was told. So Dr. Kumari is an active researcher at SOM. She has a number of grants and awards, and she's going to talk about her current research on the hedgehog family of proteins in signaling in the organ of taste. So, Dr. Kumari, please share your screen, and the floor is yours.
Hello, everyone. Can everyone see my screen?
Yes.
Yes.
Again, I apologize for this voice today. So if anything is not clear, feel free to interrupt me, and I'll repeat it and try to be clear. And thank you, Claude, for the introduction. So today I will be talking about some of my postdoctoral work and some of my recent work, and then future direction, what I'm doing right now at Rowan University. So hedgehog signaling regulation of taste organ. Why hedgehog signaling? Because it regulates tongue homeostasis or maintenance. And in contrast to maintaining tissues, if this signaling gets deregulated, it can lead to cancer. Thus, as a treatment, hedgehog pathway is inhibited. And this strategy works, but the patients who take these hedgehog pathway inhibition drugs report taste disruption. Thus, as a taste biologist, it becomes important for me to understand the mechanisms behind this taste disruption and fill this knowledge gap. So for today's talk, I'll be briefly talking about all these elements, followed by my work, which gives us some understanding that what is happening here, and then some future direction. So hedgehog signaling is a master regulator of several tissues because it is involved in the embryonic development and in the adult organ maintenance as well as in adult neurogenesis. This is a very simplified model of the hedgehog signaling, which shows two transmembrane receptors, patch 1 and smoothen. In the absence of any ligand, patch 1 inhibits smoothen and there is no signaling. But when the ligand comes and binds to the patch 1, this inhibition is relieved and smoothen can now activate the transcription factor GLE2 to transcribe the target genes, including GLE1. GLE1 is used as an indicator of hedgehog pathway activity, and I would be using this name in many of my slides. Hedgehog signaling regulate the tongue homeostasis by regulating the dynamic taste organs. And I say organs because the human tongue is a set of organs, or also known as papillae. We have three distinct taste papillae. Most of the anterior tongue is covered by these dot-like structures, which are known as fungiform papillae. At the apex of the fungiform, there is the main taste bud, which is onion-shaped, and these taste cells are connected with these nerves and pass the information to the CNS. On the back of the tongue, humans have about 8 to 12 circumvallate papillae, which are each circumvallate has two walls filled with hundreds of taste buds. On both posterior lateral sides, we have foliate papillae named because of these folded structure. And again, on the walls, we have these taste buds. In addition to these three taste papillae, bulk of the tongue is covered by the non-taste papillae, which looks conical in shape without any taste buds. And the main function is saliva movement, mastication. Here is the rodent tongue, which looks very much similar to human tongue, and that's why we use mouse model for our studies. So again, we have three distinct taste papillae. The anterior tongue is covered by the fungiform papillae, but unlike human, rodents only have one taste bud. And unlike human, rodents only have one circumvallate papilla, although both the walls are filled with taste buds. And again, on both sides, we have these folded foliate papillae, and rest of the tongue is covered by this conical filiform papillae. And here I would like to bring your attention to the GLE1 lag-Z. I mentioned the GLE1 is a target gene. Lag-Z is a reporter that is tagged to this gene. Lag-Z encodes beta-galactosidase, and in the presence of X-gal substrate, a blue-colored product is formed. So when we incubated this tongue from the GLE1 lag-Z animal in X-gal solution, blue-colored product formed only in the taste papillae and not in the non-taste papillae. This indicates that the hedgehog signaling is active in the taste papillae. I am working mainly on the anterior fungiform and posterior circumvallate papillae. And for the sake of time today, I'll be talking more about the fungiform papillae. In the fungiform papillae, the main taste tissue is this taste bud, which is connected to the exterior via these taste pore. So the stimulus is received via these taste bud cells. But these taste bud cells are not immortal. They are dynamic in nature. They turn over in a continuous fashion, and an average lifespan of the taste bud is about 10 days. So we need some progenitors for these taste bud cells, and this function is served by the perigemal cells. So in this hematoxylin and eosin staining, you can clearly make out the difference between these perigemal cells and these taste bud cells. So these perigemal cells migrate inside, they differentiate into a taste bud cells, but they are very few. We need more taste bud progenitors, and we have these basal cells that contribute to the taste bud. So this migration, this differentiation, this proliferation needs to be under tight control so that there is a continuous influx of these taste bud cells. And this tight regulation is achieved by the Hedgehog signaling. I've shown you that the Gleeman lag Z is present in the fungiform papillae, but there are more components, and I would like to show that where they are present in this fungiform papillae. So the ligand, sonic hedgehog, we used a reporter model, and we observed that the ligand is present in the taste bud and in the nerves. The GLE2 transcription activator is present in the entire basal cells, in these perigemal cells, and in these stromal cells, while the target gene is present only in the FP basal cells, perigemal, and the stromal cells. So it seems there is some kind of paracrine signaling where the ligand is here, but the signaling is active here. So the ligand must be transported up to this location. While the Hedgehog signaling is indispensable for many tissues, if it gets deregulated, it leads to tumor initiation and metastasis in more than 30% of the human cancers. One of such cancer is basal cell carcinoma, which is happened due to the Hedgehog pathway gene mutation, mainly in the patch1 and smoothen. So what does that mean? That for an active signaling, you require the ligand, but in the basal cell carcinoma, even without the ligand, this signaling remains active. Thus, to prevent this overactivation, one approach is to inhibit the smoothened so that the pathway can become inactive. But in this case, if you inhibit the smoothen, even in the presence of the ligand, the pathway will remain inactive. But we all know that FDA approves a drug when there is more benefit than risk. So FDA approved 2 drugs for metastatic or locally advanced basal cell carcinoma. Their names are vismodegib and sonidegib. And let's see how this cancer treatment is done by Hedgehog pathway inhibition. So this is a basal cell carcinoma lesion, which after 6 months of vismodegib treatment is completely gone. It's highly effective treatment. And the data shows that within 1 month, the GLE1 mRNA levels are significantly declined. That means the reduction what we see here after a month is due to the Hedgehog pathway inhibition. So Tang et al. In 2012, when he reported the efficacy of this treatment, also reported taste disturbance. In this graph, where on the x-axis, there's a duration of treatment of the drug, and on the y-axis, we have a percentage of patients without the taste disturbance. And the graph shows within a couple of months, 80% of the patients were with the taste disturbance. And similarly with the vismodegib, after a 12-month analysis, 60% of the patients report dysgeusia or taste disturbance. That means the amazing lunch or the food which we eat is experienced something like this by these patients. So if you lose a taste for a day, you can manage. If you lose taste for 1 week, which usually happen in COVID patients, they survived. But if one is not able to taste for months, it is awful. And the patient would not like to take these drugs, which can compromise the treatment success. And it also associated with the reduction in quality of life and with depression. So I wanted to understand that whether this taste disruption is a direct effect of Hedgehog pathway inhibition or an indirect effect. So my first question was, can Hedgehog inhibition by blocking smoothing affect adult taste organs? I did some morphological and functional experiments. I used sonidegib drug because of its better pharmacokinetics as compared to vismodegib in mouse model. And to be specific that the effects are due to smoothen, I used the genetic disruption of the smoothened allele. So here are my data. This is a very beautiful image of the scanning electron microscopy from a tongue from a vehicle treated group, where you can see a fungiform papilla, a pore, and surrounded by the filiform papillae. After 16 days of sonidegib treatment, this fungiform papilla is disrupted into something like this, which is significant. We confirm this finding with the hematooxylin and eosin staining. So the onion-shaped taste bud is completely lost after sonidegib treatment, and we see a thickening of the keratin layer. Next, we checked the Hedgehog signaling, that whether these effects are due to the Hedgehog pathway inhibition. So these GLI1 responding cells were completely eliminated from the epithelium and reduced from the stroma. So that means this disruption of the taste organ is due to the effect of Hedgehog pathway inhibition. So we did some quantification to understand the extent of these alterations. We categorize the fungiform papilla into 3 types. Type 1 is the typical fungiform papilla, which is rectangular in shape, having a typical taste bud, which is onion in shape with a taste pore, a flat, thin epithelium. The second category is atypical fungiform papilla, which becomes conical in shape with some atypical taste bud, which loses cells, and there is a thickening of the keratin layer. The third category is these atypical fungiform papillae without any taste buds. So I will be using these terms typical FPTB, atypical FPTB, and atypical FP no taste buds in my upcoming slides. So first I wanted to understand how rapid are the effects, because my previous data was with 16 days. So I treated the mouse with 5, 10, 16, 28, and 36 days of sonidegib. And I found that after 5 days, there are not much effect on the typical taste organs, but after 10 days, only 50% of the typical taste organs remained, which virtually eliminated after 16 days. In contrast, the atypical FP without taste bud subsequently increased, and by 36 days, the majority of the fungiform were atypical without any taste buds. When I looked at the atypical FPTB, I see that the percentage first increased and then declined. What does that mean? It means that these taste bud cells are not dying instantaneously. Instead, they are living their life cycle. They die, but they are not getting replaced by the new taste bud cells. So here we are seeing an increase in the atypical taste buds, and when the taste bud cells are completely lost, we see a decline. So to know that these effects are specific for smooth and inhibition, I genetically deleted smoothen, and I did it in two ways. In first model, I used the whole body smoothen deletion, where smoothen were deletion from epithelium, stroma, nerves, everywhere. And I observed that the data mimics sonodigib, where there is a elimination of typical taste organs with subsequent increase in atypical FP no taste bud. And here I would like to remind you that when we looked at the Gleason responding cells, we saw that it was eliminated from the epithelium and some of the stromal expression were there. So in my next model, I eliminated the smoothen only from the epithelium. But the data showed similar results as that of sonodigib, that after 5 days, there is not much reduction in typical organs, which reduced after 16 days, and there is an increase in the atypical taste organs. So that means the epithelial hedgehog signaling is the main contribution for these taste buds, and these are the taste bud progenitors cells. So next, I wanted to look at if any other taste organ, because we have 3 different taste papillae. So I looked at the posterior circumvallate papillae, and in the vehicle, you can see these oval-shaped taste bud, which after sonodigib treatment is eliminated. You can see these empty spaces, and the same was observed in both of the smooth and deletion model. We can see these empty spaces. So for quantifying this data, I measured the taste pore. So you can easily distinguish the taste pore from these taste buds as compared to these taste buds. So when we quantified, and I'm showing you just the data obtained from whole body smooth and knockout deletion, that there is a significant reduction in the taste bud pores. So this sonodigib is affecting both anterior and posterior. And the rest of the data you can see in this paper if you're interested. Next, I wanted to understand about the innovation, because if the innovation is required to transport the chemosensory information from the periphery to the central nervous system. And in the fungiform papillae, there are 2 nerves. Lingual nerve is a nerve that innervates the entire tongue, the filiform and the fungiform. The lingual nerve enters into the fungiform, but not into the taste bud. It is this cauda tympani taste nerve which enters inside, and we have recently observed that it is outside, lining the taste bud. So we have antibodies to label these nerves. Lingual nerve is measured by the, indicated by the neural filament, and K8 is labels the taste bud. And we can see that after 28 days, without any taste bud, these innervation is retained. So the overall innervation was intact. Let's see the specific innervation to taste bud. And you can, I hope you can see that there are fibers outside the taste bud. So what we have seen that after 28 days sonidegib, even with those taste bud remnants, the cauda tympani was also intact. So this was great because if these nerves were intact, we can do the nerve recordings. And because this cauda tympani nerve innervate these taste buds and pass this information to the CNS, we can cut this nerve, put it on the electrode, and do the nerve recording. So as we flow the taste stimuli, we get the signals. So the way we do it, we put the stimulus for 20 seconds, followed by a rinse, second stimulus, rinse, and so on. And for quantification, we integrate this data, and the height is measured as the response units. For this experiment, we have used chemicals that represent salt, sour, sweet, bitter, and umami, all the 5 tastes that we can perceive. In addition to taste, we can also feel the touch and the temperature. So I have included these stimuli into my experiment. So first, let's look at the chemosensation. So when we recorded from the cauda tympani in a vehicle, we see responses like this, and you can see that different concentration of sodium chloride elicits different height of response because we have the ability to detect different strength of the chemicals or the stimulus. But after 16 days of sonidegib treatment, all the responses were lost. And as we anticipate, after 28 days, there were no responses to any of the taste and chemical stimuli what we used. When we looked at the somatosensation, and we used, as I said, we use the touch, and the way we do it, we stroke the tongue 1, 2, 3, 4, and 5 times. And because the nature of the response is rapidly adapting, we do not integrate this data. And we saw that after 16 days of sonidegib, this stroking elicits response. That means even though the chemosensation was lost, the somatosensation was retained for both touch and the temperature. So it was interesting to us because the taste bud receptors were lost. So who is giving response to these touch? So we know that the nerves were retained, and the specific nerve to the taste, which is indicated here by the FOX2B. So we use the FOX2B as a marker for cauda tympani instead of using the P2X3 antibodies, because the reporter model always works better than the antibodies. So what we did, we looked at the FOX2B perigemal projections. Because we recorded from this nerve and the taste buds were lost, we were interested to see that whether these perigemal projections are responding to the touch sensation. So we found different kind of projections. And we made the hypothesis that there are 2 categories of this cauda tympani nerve, one that enters into the taste bud and one which is lines the taste buds, the perigemal projections. And after sonidegib treatment, we found that all these perigemal projections were there even though there were no taste buds. And the second interesting finding was those intragemal projections, they were not eliminated. They were there, they were just remodeled. This is our very recent observation we published in December 2021. And we made a suggestion that these chorda tympani nerves are responding to touch and not those cells. Those cells would be responsible for the chemosensation, but the somatosensation is achieved by these nerves, although we do not rule out that these entrachemal projections can also sense the touch sensation. So next, we wanted to learn that whether discontinuation of this drug leads to taste recovery, because this is given to patients and the taste recovery is an important aspect. So I did the experiment in my approach was to do the recovery after short-term pathway inhibition, because after 16 days, all those 3 types of fungiform papilla were apparent. So after 16 days of treatment, I discontinued the drug treatment and animals were maintained in the standard housing for 7 days until 9 months. In my second experiment, I did a long-term hedgehog pathway inhibition of 48 days because at this duration, most of the fungiform papillae were atypical without any taste buds. And again, I did a similar recovery for 5 to 7 months and analyzed both the taste organ and the function. But for the sake of time today, I'll just show the taste organ data. So is recovery possible? And we did find that a subset of fungiform papilla recovered. So the typical taste organs, which were almost eliminated after 16 days, after a week of recovery, we did not find much of the regeneration, but after 14 days, 50% of the typical FPTB were regenerated. But interestingly, even after 9 months of recovery, this percentage did not increase further. When we looked at the atypical FPTB, after 7 days of recovery, the percentage reduced and further declined after 14 days and were comparable to vehicle for 9 months. That means what did not recover was this category, atypical FP, no taste bud. So after 16 days, there was an increase as compared to the vehicle. After 7 days, there is further increase. That means there is a lag period where once you stop the drug treatment, it did not, the increase was continuous. And after 7 days, there was, this percentage was maintained up till 9 months. So for some reason, this category was unable to regenerate. When we took at the long-term hedgehog pathway inhibition, after 48 days, most of the fungiform were without any taste bud, and as we would anticipate, this category did not recover. And this is just the graphical representation of the same. So when I came here, I wanted to understand why this papillae did not recover. And I observed because the hedgehog signaling was not resumed in these atypical papillae without any taste buds. And why the hedgehog signaling was unable to resume here, because of this hedgehog pathway antagonist, HIP, we found that this HIP is present only in the filiform in the control animals, but after hedgehog pathway inhibition, there is ectopic expression of this HIP molecule in the fungiform. In these atypical FP taste buds, there are some taste buds and the associated sonic hedgehog ligand is available. So when we discontinued the treatment, somehow this sonic hedgehog was able to eliminate this H-hip and resume the hedgehog signaling. But in this category, where there is no ligand, stopping the treatment even for 9 months were not able to inhibit the H-hip, and this category cannot recover. So we published just last month this observation. So our data suggests that this taste disruption is an on-target effect of the hedgehog pathway inhibition. So I would like to summarize these observations with the implication for patients. So my data showed that the hedgehog smoothen inhibition disrupt the taste organ. That means the patients who are on the hedgehog pathway inhibitor drugs will experience taste deficits. Taste bud defects were observed in both fungiform and circumvallate papillae. So the patients can expect a whole mouth aberrant taste effects. We also saw that the taste organ disruption is duration dependent. That means this disruption in the taste responses reflects a significant taste bud loss. Loss of taste responses even to the high concentration of chemical stimuli. That means even the strong taste stimuli is unlikely to elicit any taste response in these sonidizib treated patients. Touch and cold sensations were retained. So the patient's diet should focus on texture and temperature characteristic of nutrients. And these cancer patients were sometimes given ice chips to motivate them to eat, even though they cannot sense the chemo sensation. With the cessation of drug treatment, we observed that the long-term exposure prevents the restoration, while the short-term leads to incomplete recovery. So if possible, having some frequent breaks during the treatment to avoid the long-term exposure might be effective to ameliorate the taste bud loss. So now what I am trying to do next. So we know that the hedgehog signaling regulates the fungiform papillae. So I made a little schematic to show how heterogeneous and complex this organ is. It has this basal layer, suprabasal layer, keratin layers, taste bud cells, perigemal, stromal, nerves. But I just want to bring your attention back to the hedgehog signaling, which is active only in the fungiform papillae. We know this because of the presence of the target gene, whose expression stops at the base of the fungiform papillae as if there is a stop sign. But interestingly, the GLI2 transcription factor is beyond this stop sign. When the pathway is active only in the fungiform papillae, why the transcription factor is expressed in these non-taste papillae? That means we have two distinct epithelium, taste and non-taste. And I'm interested to delineate the differential regulation. So it is clear that these cells are giving rise to the filiform and these cells are giving rise to the taste bud, but the experimental evidences are lacking. So what I'm trying to do here, I'm trying to utilize a GLI1 reporter model because this model allows me to label the taste epithelium. I inject the tongue with the enzyme so that I can separate the epithelium, because right now I'm interested only in the epithelium. I prepare a single cell suspension and I stain with a basal layer marker because the basal epithelium is the location where the GLI2 is expressed. And then I run this suspension through fluorescence activated cell sorting. And I sort the taste epithelial cells and the non-taste epithelial cells. The extra cells are just discarded. And the taste and non-taste epithelial cells are used for RNA isolation, library preparation, sequencing. I'm working with the Dr. Benjamin Caron group to understand what is the transcriptomic difference between this population and this population. So GLI2 is a target gene. My another set of experiment is what are the target genes in this population and in the non-taste populations. For that, I'm using the chromatin immunoprecipitation. So how this technique works, GLI2 is a transcription factor, so it is attached to the DNA. I fixed the cells, so the GLI2 becomes attached to the DNA, and then in subsequent process, I just concentrate the GLI2, and in the final process, I unlink the GLI2 so that I can isolate the DNA, and the DNA will get sequenced. And we will analyze what are the target genes in the taste epithelium versus the non-taste epithelium. So this information would be helpful to compare that if those target genes should also present here. So this is first experiment. My another experiment is, we know that after sonidizib treatment, the taste buds are lost. The taste progenitors were still there. We confirmed that the hedgehog responding cells were not there. But when I looked at the GLI2 lag Z, the GLI2 lag Z were still expressing after the sonidizib treatment, even in these atypical fungiform papillae. So if the GLI2 is not transcribing this GLI1, what it is doing here? My hypothesis is that GLI2 might be responsible for transcribing the genes like HIP. And after the hedgehog pathway inhibition, the HIP is expressed here. So maybe the target gene has changed after hedgehog pathway inhibition. So what I'm trying to do is to know the genetic and molecular alteration in the lingual epithelium after the pharmacological hedgehog pathway inhibition. And my approach is, again, utilizing a GLI1 mouse model, but this time this mouse model gets treated with the sonidizib. And I take the epithelium out, prepare a single cell suspension, sort these cells into taste and non-taste. RNA will be extracted for the transcriptomic analysis, and GLI2 chip will be done to determine the GLI2 target genes after the hedgehog pathway inhibition. In addition to these advanced studies, I'm also doing some basic tongue studies. I'm using a mouse model, and I harvest the tongue from different mouse models. I do cryosectioning and then histological analysis of the tissue, as well as immunofluorescence staining of the tissues to understand, like Ki67 is for proliferation, for the taste bud, for the nerves. So which model? So there is one observation that in this Niemann-Pick C1 disease, hedgehog signaling is deregulated. So Dr. Ileana Soto is working in this mouse model, and she generously agreed to share this mouse model with us. So we are trying to study that whether in this model the hedgehog signaling is disrupted. If disrupted, can we see the effects, what we have seen with the sonodigit? In addition, there are reports that the Alzheimer patients also have some alteration in the taste. So one of faculty in Rowan SOM is working on the Alzheimer rats. So I have taken rat tissues from those Alzheimer rats, and I'm also trying to analyze using this strategy. And I'm also looking for those cancer drugs like taxane-based, which also reports taste alteration. So this I'm doing in my mouse model. One of the taxane-based drug is docetaxel. So I'm using my Gleeman-Laxi model. I treat the drug for different duration, and then I harvest the drug, and then I do these analysis. So once I'll have the data from the basic studies, we'll know that how far we need to go to understand whether the mechanism or what is happening. So with this, I would like to acknowledge and my postdoctoral lab, Ms. Shweta and Bradley lab. Libo was our tech and support. And SUK and Hajime have done those neurophysiological nerve recordings. And I got a picture of Hedgehog because these labs provided me support for the Hedgehog signaling model. I got the smoothen model from the Delagoche lab, and Ellen lab has provided me the chip-related models. And currently I have some students who have done last summer some short-term projects with me. Some Lion and Javaria are currently working with me. Magdalena and Rashmi were lab technician with me, but they had to leave because of some personal issues. So I am looking for a lab technician right now. With this, I would like to acknowledge the funding, the R01 to these PI and R21 to me that helped me to continue my studies here. With this, I would like to take any questions if you have.
Well, thank you very much, Dr. Kumari, for this very interesting talk. I really learned a lot about my tongue and obviously the importance of taste. So I think we're going to, I'm going to ask you some questions that were curated by Dr. Eaton from what was posted on the chat of Zoom. And then we're going to take maybe another 5 to 10 minutes to listen to you talking a little bit about your career path, you know, and what you would recommend to students who are undergrads, what they should do if they want to become a tongue scientist or hedgehog scientist, the way you are. So there are a few questions. Some of them are more or less generic. I will start by selfishly by one of my own questions, because as a virologist, I cannot help myself. And you mentioned that one of the symptoms of COVID-19 is actually a loss of smell and a loss of taste. Do you have any idea if there is any knowledge of why taste is lost during a COVID-19 infection, if cells are lost, papillae are lost, or just the signaling?
So the signaling is intact. So this virus needs a receptor ACE2, which is shown to present in the lingual epithelium, and the research is still being continuous. And recently it has been reported that the taste bud cells are also being lost, but it is not due to the hedgehog signaling. Hedgehog signaling is separate. Because of the presence of the ACE2 receptor in the tongue, there is a direct effect on the tongue.
Oh, okay. And another questions about your knockout model. You have a whole body smoothing knockout model, and hedgehog signaling if I remember well, is very important during development. And
Yes.
so I wonder if there is actually a more generic effect on these smoothing knockout mice beyond just the taste organs.
So it did affect hairs, and Dlugosch lab was working on the hairs, and but this model was conditional deletion. The smoothened model, if the smoothened is deleted from birth, the animal won't be delivered. So that's why we use the conditional deletion model. So in adult, we deleted for 5, 16, and 24 days, which were the animals survived well, but they did have some like abnormalities in the hairs, which is all together different, and I did not analyze any other tissues. So I'm sure about the hairs alteration, but not sure about any other alteration.
Oh yeah, that makes a lot of sense. Another question from Dr. Soto. She said that it was really a beautiful work, and she asked if there are specific inhibitor for the transcription of GLI2.
There are, and in fact, these smoothened inhibitors are now showing resistance. And there are some BET chromodomain inhibitors which inhibit the GLI2. So these GLI2 inhibitors, there are like GANT inhibitors which directly affect on GLI2, and now they are under clinical trials for their efficacy.
Oh, I see. Yeah.
And we have also worked on the GLI2 genetic deletion, and we have shown that in addition to the smoothened inhibition, if GLI2 is also inhibited, we will observe similar loss of taste organ alteration and taste loss.
Okay, so because there are other forms of loss of taste, like parosema, I think the name is, of losing taste. If essentially, are there other ways for us to lose taste other than these specific inhibitors?
Yes, so there are like geographical tongue where the location of the taste bud is changing. So there are local effects on the tongue which can lead to taste loss. Yes.
And
there are
since,
like burning mouth syndrome, and there are different symptoms.
so you're talking about geographic tongue, which is a remarkable name and appearance of the tongue of people affected. And the question is, you show all these taste buds. Are they all sort of generic taste buds that taste all 5 tastes, or do we have specialized taste buds for
Yes.
sweet, sour, and so on?
Yes, I'm glad you asked. Yes, there are heterogeneous taste bud cells. We have so far identified 3 different types of taste bud cells. Type 1 is reported for salt sensation. Type 2 is reported for sweet, bitter, and umami. And type 3 is reported for sour. I did not show just for the time's sake, but I did analyze all those 3 types in my studies, and I have seen that all those 3 types were reduced. So it was not one single type which was more affected than the other. Once the hedgehog pathway is inhibited, all the taste cell types were affected, and that's why I think we have seen the elimination of all the taste responses.
Yeah. You showed that you can actually define by stimulating taste buds with specific chemicals. You can define the nerve response, so it gives you an idea whether these cells are actually functional.
Yes.
But in the case of a mouse, when you treat these mice with these chemicals, can you tell if the mouse has altered taste based on whether they eat more, they eat less, or they feel depressed, or they don't look for food, or they lose weight, or can you see something happening to them?
So we did look at the weight, but it was a 16-day experiment, and within 16 days, there was no weight changes. We did look at the weight at 48 days, but it was a 48 days of continuous treating the animals with an oral gavage that goes via the mouth. So the weight declined both in vehicle and the treatment. So we looked at the weight. There are other ways of doing behavioral assays. There are some crude, like 2-bottle test, where you can give salt, sucrose, and the quinine. So you will anticipate that if the mouse has an intact taste, it will take the sucrose but not the quinine. And if it doesn't have the intact taste, it can take the quinine also. But for our studies, because we have done the electrophysiological recordings, we have done a surgery, and it was a non-recovery surgery. So the animal had, we could not do both the recording and the behavioral taste responses. And because we were doing the uh, when we record, we have the ability to do all these touch, temperature, and all those 5 taste stimulation at the same time. We found that it is more powerful than doing a behavioral essay, because the behavioral essay is just a reflection of the taste responses, what we see.
All right, thank you. So I'm going to switch to a slightly different question now. You mentioned that you're looking for a lab tech. What are you looking for when you hire somebody for that particular position? And could an undergrad or recent graduate apply? And if
Yeah,
so,
definitely.
how should they reach you?
So I think the posting is available in the Rowan career website. They can apply through that. And I believe Tang is like a simple model to work with. So I train these students. So even the fresh graduates, I am willing to take them, and I can train them how to do cryosectioning, how to do harvesting, how to do staining, imaging, and then the analysis. So right now I'm not looking someone with having those sequencing and the bioinformatics analysis. That is a plus. But even with a fresh graduate without any experience, yes, can apply.
Okay, so yeah, I encourage people who will graduate soon to apply. I mean, um, and I would like you to take another 5 minutes, a little bit or more, to talk a little bit about your career, what makes you decide to become a scientist, and how you got to your current position, and what you think was very helpful and maybe not so helpful during your career.
Sure. I can stop sharing this. Is that okay?
Oh, yes.
Okay. So as you mentioned that I have done my initial studies in India. So while doing my PhD, I was doing the reproductive toxicology, and I used a mixture which has the chewing tobacco mixture. And I saw that the reproductive toxic effects were transmitting through from first generation to the next generation. So I was very interested to do some epigenetics. So I came to US to learn more about the tools and techniques about the epigenetics at University of Michigan. But after 1 year of work, there were some funding issues that was very bad. It wasn't a year that I had to leave from that lab. And I tried to find another lab with the same interest with the epigenetics. But that year, I think 2012 was not a great year. There was NIH funding cut and what's not. So there was not much of the opportunity available. So when I looked at this tongue and taste, I always wanted to work with something which has a translational value, like a not like a drug development, which can take up to years, but which we can understand a quicker way. And that's why, like, I started my PhD in that reproductive disorders, because infertility is quite an issue. So when I looked at the opportunity in that taste field. It was a small field. Not many people work on the taste, but I was interested because of the translational value and because of the developmental biology aspect. The lab was working on developmental, uh, the tongue development in the context of hedgehog signaling, and recently started looking into the adult tongue. So when I joined in, uh, as I said, in 2012, there was a report about this taste alteration. So when I joined the lab, we initiated this study about that what happens in the taste organ or the non-taste organ. We saw that the taste organ was affected, but not the non-taste organ. So there, slowly we built up that, okay, if, like you said, that we first started with smoothen, we turned into GLE2. Because mouse was a small model, I did some study on the rats, and I published that the taste signaling generalized to rat. Whatever I have seen in mouse was similar in rats. And the purpose I did in rats, because I wanted to, I have looked at the tongue, I have looked at the innervation. Now the final part is the central nervous system, which is more accessible, and we can do more studies in the rats. Right now, those studies are in progress that shows that in addition to the peripheral disruption after the sonodigib drug, there is central disruption also. And in the University of Michigan, I learned to collaborate. I collaborated for my different models. I collaborated for my rat models, and I collaborated for my electrophysiological recordings. So I will encourage all the upcoming researchers that we cannot do everything by ourselves. And if we collaborate, we do better, we do faster in an effective way. So I think in these 5, 7 years, whatever data I have accumulated, the collaboration really helped me. And those collaborations also helped me to provide the letter of support while I was providing my grant. I also received a small grant from the University of Michigan. And again, those collaboration helped me because the people knew me that I'm doing those work. So right now, when I came here, I came just before pandemic. It was the hardest time. I was not able to get these mouse models, and it took me a long time to take extra hands and people. But when I started to uh, like, and as I said that uh, that grant helped me to come here. That so I think uh, if I wouldn't have that grant, I don't know, like if I would have been here. So when I came here, I wanted to again collaborate with my colleagues out here. So as I mentioned that the bioinformatics analysis I'm doing with Dr. Ben, and one of the mouse models I got from Dr. Ileana Soto. And I'm not aware what is happening in rest of the departments, but if they feel that tongue could be looked upon, I'm open to collaboration and look at the tissue.
Yeah, so I totally agree with you. Collaboration is absolutely critical now throughout, and I think it's great that you've already established these collaborations throughout these.
I'm great. I'm thankful to my colleagues who agreed to collaborate.
Somebody asked here that when you were a child, did you know you were going to be a scientist? Did you?
No.
Be a scientist?
What I want, sorry, I interrupted you. Please finish your question.
No, it's okay. That was it. That was the question.
Okay. Uh, so my grandfather was a doctor, and I've always seen him treating patients. So I was inclined towards working for for the human life. So when I started in my when I finished my schooling, I wanted to learn about all these organisms which are causing the disease. So I did my undergrad in microbiology. And I understood that I cannot do all. It's so much. There are 5 types of microorganisms, but it was 3 years that I learned them. So then I switched myself to learn the tools and techniques which I can use. So in my master's, I have done biotechnology. And once I understood that what I have, I started my studies in malaria because that was also an issue. So I always knew that I wanted to do something related to human disease or disorders. But as a child, I was not sure whether I would become a physician or I would become a researcher. As I did my studies, I thought by doing research, I have more opportunities to discover, to learn, and to disseminate the information.
I think this is something that a lot of us faculty researchers and students actually share, that but what you become, what you decide to be is also always changing and evolving based on opportunities. And as you said also, the influence of funding, the influence of opportunities make you sometimes change your field of research, and
Yes.
it's very often for the best. And so I think we will finish here. I want to thank you again for this very, very interesting presentation.
My pleasure.
Very clear and perfect for our biology and biomedical students here. So
Thank you.
thank you very much. And I remind all the audience that there are upcoming presentations in our biology seminars. And again, on March 10 is our next presentation. So please look at our schedule and join us for our next presentation. So thank you again, Dr. Kumari, for that excellent talk.
Thank you for bearing my voice.
It was very good.